It is time for us to get into the other part of the business. It gives me a great pleasure to welcome Dr. K.C. Narayan Swamy, Director of Education, USB, for the opening remarks. Dr. K.C. Narayan Swamy is the professor of sericulture and he is well known for his work on silkworm oozy fly. Very recently, in a collaborative research, we generated useful information on growth and development of mealybugs in transgenic mulberry. As a director of education, he is providing direction for improving the quality education at our university. I request you, sir, to deliver opening remarks.
Thank you. Thank you, Dr. Karba. Good morning to each one of you, respected Vice Chancellor and President of the today's inaugural session, Dr. S. Rajendra Prasad sir, distinguished scientists, chief guests of inaugural session, Dr. Subrai Hegde, USDA, respected Dean PGS, Dr. N. Srinivas sir, who is the person behind this wonderful program, my colleague Dr. N.B. Prakash, Dean Agri, College of Agriculture, GKVK, distinguished speakers, eminent scientists, deans of constituent colleges of UAS Bangalore, officers of UAS Bangalore, heads of the departments, faculty members, PG students, invitees and friends. I am very happy to note that UASB is organizing 8th series, that is the 8th edition of Science Week for 4 days from 27th to 30th September 2020. So involving distinguished speakers distinguished speakers as well as alumni of UAS Bangalore from different parts of the world. I once again wish you all a very happy Science Week 2021. So before I start my remarks, opening remarks on Science Week, I would like to say a beautiful statement by Albert Gent. Research is seeing what everybody else has seen and thinking what nobody else has thought. Research is creative and systematic work undertaken to increase the stock of knowledge. It involves collection, organization, analysis of information to increase the understanding of a topic or issue of our interest depending upon the situation. The primary goals of the research are there is a researcher within all of us, but to come out with flying colors, a good researcher must be competent enough to discover something. He must be skillful, strive for high quality, high standard research, and research requires a critical analysis, but most of all, common sense. Qualities of a good researcher are possessing analytical mind, intelligence, curiosity, quick thinking capacity, commitment, determination, excellent written and communication skills, and doing the work very systematically. This is the most important. The meticulous, conduct of experiment, whether they are social sciences, whether they are experimental sciences or biological sciences, one should conduct the research very meticulously and systematically. In addition to these qualities, a researcher must have ethics and provides guidelines for conducting the research responsibly. In addition, it educates and monitors scientists conducting research to ensure high ethical methods or standards. Important research ethical principles are honesty. One must be honest to the subject, objectivity, integrity. Integrity is also very important. Carefulness, openness, respect for intellectual properties, confidentiality, responsible publications, responsible monitoring or mentoring, social responsibility, non-discrimination, and legality. These are the important ethical principles of any researcher. A great work are performed not by strength but by determination. One should keep it in mind. It is essential to understand and avoid research misconducts like fabrication of data or fabrication of theses, falsification of data, falsification of experiments, and most importantly, plagiarism. Research misconduct does not include honest error or differences of opinion. Researchers have different tools that they are based on what type of research they are going to conduct. For example, interviews for qualitative research, experiments for the quantitative research. So this is also important. Based on the problem of research, use an appropriate tool, put an insight to your thinking through necessary action and ethics, and publish your findings for the benefit of other researchers as well as for the benefit of society. Publishing is one of the necessary steps embedded in the scientific research process. It also necessary for graduation. See, in fact, the UAS Bangalore has made it compulsory to publish at least two research papers for submission of thesis. So the publications are very important for graduation and also for career progression. Publish new and original results and methods only. Reviews and summaries of particular subject. Manuscripts that advances the knowledge understanding in a certain scientific field. Do not publish results of no scientific interest, outdated work or outdated research, duplication of previously published work, and also please do not publish incorrect or invalid or unacceptable conclusions. While selecting the journals to publish your work, ask following questions yourself: whether the journals are peer reviewed. So we have to question
Recording
ourselves:
stopped.
who is this journal's audience? Who are the referees or who is referring these journals? What is the average time to
Recording in
print or publish
progress.
the papers? Then what is the journal impact factor? You know, the journal impact factor is generally measured in terms of number of times the citations are being made with respect to a particular article or the published paper. Publish the articles in journals that are peer reviewed and have high impact factor. You know the importance of the impact factor. Wherever you go for interviews for the higher studies, they ask for the publications with the high rated journals and high impact factor. See, I would like to mention here three important aspects in a published research paper. They include: the first part includes title, abstract, and keywords. Make them easy for indexing, understanding, and searching. It must be informative, attractive, and also effective. The second part includes main text, that is introduction, methodology, results, and discussion. Results and discussion. Journal space is not unlimited. Please keep it in our mind. Journal space is not unlimited, but your reader's time is very important, that is scarce. But your article as concise as possible. Make your article as effective as possible. The last part of the research paper is conclusion, acknowledgement, references, and supplementary data. Mention most important outcome of the article. So we have to mention the most important, application part or conclusion of a research article at the end of the research. Important points to remember while publishing the research paper are: do not spend too much time on your preparations. Submit to the right journal, submit to the right journal only. Check the English, pay attention towards the quality, revise it several times, show it to your mentors, and understand the journal's requirement, and be honest in preparation of research paper. So with this, in fact, I have gone through the program drafted for the purpose. In fact, there are excellent renowned speakers for about 4 days. This has been structured very well. There are 104 PhDs. Students will be delivering their research topics in four concurrent sessions. So it will be systematically evaluated by the eminent faculty members from outside the university and also from within the university. So at the end, I would like to say that no research without action, no action without research. So this is well said by Kurat Levin, a German American psychologist known as one of the modern pioneers of social, organizational, and applied psychology in the USA. So I congratulate the organizers for having drawn the wonderful program for 4 days. So I request all the faculty members, all the PG students to, effectively utilize the, the schedule, effectively utilize the schedule and interact with the speakers for enriching the knowledge in the respective field. Thank you, sir. Thank you very much for the opportunity. I thank all the team members for wonderful organization. Thank you.
Thank you, sir.
Thank you very much, sir, for your advice. I agree with you that disciplined manner research can help us to do wonderful contribution for the database of knowledge. Thank you so much, sir. So with this, now we will move on to other component of our major activity today, that is keynote address. So it gives me a great pleasure to introduce our keynote speaker, Dr. Subrai Hegde, Director of Biotechnology Risk Analysis Programs, USA. So before I request him, I just want to read his biodata for the benefit of the students listening to this particular session. Dr. Hegde is currently the Director of Biotechnology Risk Analysis Programs, a unit within the Biotechnology Regulatory Services of Animal and Plant Health Inspection Service, they call APHIS, of the United States Department of Agriculture, USDA. His program is responsible for authorizing permits for interstate movement, importation, and field testing of organisms developed using genetic engineering and approving petitions for deregulation. He is actively involved in international capacity building activity in biotechnology regulations and traveled extensively in South and Central America, Mexico, Canada, Europe, and Asia. He is currently a member of the Organization for Economic Cooperation and Development, OECD working group on the harmonization of regulatory oversight in biotechnology. In 2020, Dr. Hegde was awarded the USDA's APHIS Scientist of the Year award for leading a team of scientists to issue a permit to test the Florida-wide release of genetically engineered virus to control devastating citrus disease, as our dean mentioned, known as citrus greening. Dr. Hegde is one of the authors of USDA's revised biotechnology regulations implemented in the year 2020. For their contribution towards the revised biotechnology regulations, Dr. Hegde and his team were recognized by USDA in 2020 with its highest award, the USDA Secretary's Honor Award. Congratulations to you, sir. Earlier, Dr. Hegde worked as a research scientist at the University of California, Riverside, and published several research papers on population genetics of wild relatives of croplands and genetic basis of wheat evolution. He obtained his BSc Agriculture, MSc Agri, and PhD in plant breeding and genetics from the University of Agricultural Sciences, Bangalore. For his MSc and PhD research project, he investigated quantitative genetic changes in groundnut and rice subjected to multi-trait index selection. Both as a tenured and as an adjunct faculty, he taught classes in plant breeding methods, molecular genetics, population genetics at both public and private institutions in the United States, also at US Bangalore. I am very happy to say that I am one among the proud or lucky students of Dr. Hegde. Dr. Hegde is a truly homegrown scientist with global recognition. With this background, it's time for me to request Dr. Hegde to deliver keynote address.
Sir, thank you, Karaba. Can I share my
Right,
screen?
sir. You can share your screen and talk, sir. Yes, sir.
Okay.
Please let me know, sir, if you have difficulty in sharing screen. Yes, it is coming.
Can you see it?
That's we can.
Okay, thanks. Thank you, Karaba, for the wonderful introduction. I'm extremely, honored to, when you guys invited, because I'm one of the alumni. And I'm really proud of our education in the University of Agricultural Sciences. So whatever we are achieving is because of that good education we received. I'm University of Agricultural Sciences. Thank you, Srinivas. He's my classmate, and also Ravi Kumar, Karaba, and all other people who really invited me to give this keynote address. It's really an honor. And also, I think Narayan Swami gave a extremely nice address regarding the value of science, how to be a good scientist. And thank you to all the organizers for this invitation, and also the students who are going to listen to my presentation. This is a science week, okay, and I'm giving certain examples here in my career, how we use science in regulation. But what example I use is immaterial. But what is important is how you use the best science in your career, how you become a good scientist. So that's what I'm going to talk. Though I know most of the examples are plant breeding, but you should not think that is the main key point here. But the key point is how you become a good scientist and use science-based, what you call an evidence-based approach in your everyday research in your career. Okay. Today, I'm talking about science-based biotechnology regulation, a United States perspective. So why I'm talking about US perspective is, can you hear me?
Yes, sir. We can. Sir, I request others kindly to mute your microphone, please. Thank you. Sir, we are connected. No problem, sir. Thank you.
I'm saying US perspective doesn't mean that is the right way of doing it. Each country has its own regulation based on what they want to protect. So I'm not really, imposing, okay, this is the way to do certain things, but I'm just giving how we have done it. So if there are any lessons you can learn, that would be great. Okay. So I have, there are 4 topics I'm going to cover during next 30 or 35 minutes. The first one is milestones in plant breeding. Second one is genetically engineered plants, science-based regulations, what lessons we learned, and future regulations.
Sir, is it possible to make Hegde sir slideshow? If it is not, we can go like this, no problem.
No, I think, I'm using Mac. It's not allowing me.
No problem, sir. Yeah, no problem, sir. We can go. No issue, sir. Go ahead, sir. No. Sorry. Yeah.
Is it possible for you to then make it bigger?
I can use the screen here, but then it is difficult to connect and reconnect. Okay. It's okay, sir. Go ahead, sir. Sorry.
Okay, Dinesh.
Yeah, I hear some background noise here.
Okay. We are extremely sorry. I'll request again all the delegates, participants, please mute so that we'll not have feedback. Sorry for the inconvenience, sir. Go ahead, sir.
Yeah, that's okay. Yeah, if you look at the milestones in plant breeding, so it is a very busy slide, but you don't need to worry reading everything. What I would like to say is, 10,000 years ago, agriculture revolution started in the Middle East. Then there are so many progress every 100 years. And in 1859, I believe, Mendel finally, discovered, the genetic basis of improvement. Then we have tissue culture. We started doing an improvement through tissue culture. Then we have genetically engineered plants in 1970s, 1990s. And now we have reached genome edited crops. So what I mean to say is last 10,000 years, we have been improving plants. But remember, for most of the time, during millennia, we did not have any regulations. So we only started biotechnology regulation in 80s. So I just wanted to know why this regulation came in the first place, where people are eating food and they considered it is safe, but we entered the regulating plant breeding, regulating crop improvement. Okay. So I just wanted to show that some of the greatest revolution in plant breeding is in around 1926 and in United States. So they started producing hybrids in corn. Suddenly there is a tremendous increase in yield. You always remember food security, the most important thing for every country. So this is the major milestone in crop breeding. Then in 1940s, mutagenesis, that is, the scientists started using X-rays, gamma rays, and chemicals to mutagenize plants and looking for the useful traits. That is another milestone in agriculture. But, I have put a slide there. If you look at in 1994, genetically modified organism or GE, so there we started regulation. Regulation was not there before. And now at the end, we have the genome edited crops. People are debating everywhere, including India, how to regulate such plants. Okay, why we started regulation? There must be a reason, right? In science, regulating genetically engineered plants, how it got started in the first place. Earlier, people used to just select best plants from ragi, for example, or rice. Whatever phenotype they found it is useful, they selected. Or then they wanted to cross between, let us say, different species of ragi to see whether they can combine good characters between plants. But what happened in 1970s, in 50s, 60s, first time the scientists can create, a new organism by combining genetic material from different organisms, plant and a bacteria, plant and animal. So that is the major change in science, that is combining things from entirely different organisms, from different kingdom. For example, Paul Berg, who is the Nobel laureate, a professor in Stanford University, first time created a recombination between a monkey virus, which causes cancer, and another virus called bacteriophage lambda. And he wanted to put that one in E. coli. E. coli is in our gut. We need this bacteria in our gut for digestion. Now, suddenly other scientists are worried. What if this microbe, this recombinant DNA escapes to human being and may cause cancer? So that means there is a risk. So this was the first time, in crop breeding. Okay, you need a regulation to prevent risk or hazard. So that was the genesis of regulation in crop improvement. If you come to United States and agriculture, so here are some of the major revolution in genetic engineering. So Watson and Crick in 1953 discovered DNA double helix. 1972, recombinant DNA bacteria was created. In 1983, recombinant DNA in tobacco plant was created. So because of all these things, people are worried. Okay, is it going to be a big risk in the environment and human health? So in 1987, US Department of Agriculture came out with a biotechnology regulation because US was a pioneer in genetic engineering. So it was the first country coming out with a regulation. In 2020, we revised that regulation, which I will touch upon later. Okay, what is important here is evaluating safety, balancing innovation with safety. Okay, why we have regulation? You use cell phone, you drive car, you drive motorcycle, and there is always a role for government. Is it safe? Is it safe for the people? Is it safe for the environment? So that is the government role. So in case of biotech regulation in the United States and the agency which I work for, we are worried always about plant health. By genetically engineering, are we affecting plant health and the environment? And there is a Food and Drug Administration, looks at food safety. And there is an Environmental Protection Agency, looks at if you produce pesticide in plant, is it safe for the environment? Okay. So in 1987, the concern was, so genetically engineered plants were created using plant pests, like Agrobacterium tumefaciens is a plant pest. It causes tumor in plants. And they were using sequences from cauliflower mosaic virus as a promoter or a NOS terminator. These are the things required to express the gene in a plant or in any organism, for example. So people are worried that time. What if you use a pest sequence, would it makes a plant pest? Does it increase the pest, or create new novel pest? Oh, people are really worried. So that's why we had a regulation regarding whether this using plant pest, are we going to create a big problem for agriculture and the environment? Okay, now I come into the risk assessment. Okay, now the science-based, we said, right? Whatever we do is science-based. What is a risk? So risk is, let us say there is a harm. Okay, like in a cell phone, for example, you use. Okay, now cell phone, let us say there is a harm because it has some kind of radiation. Now, you do not get harm if you don't use cell phone at all. But if you use cell phone, that is the exposure. You are exposing yourself to the cell phone. So now you can have a risk. But there are some uncertainties because there are so many publications. Some people say there is no risk. Some people say there is some risk. So it's always, this is the simplest risk equation. In general, risk is referred to the likelihood of an adverse outcome. That is, it is a likelihood. Remember, in science, you do not know 100%. See, because science does not talk about truth. Science is always talks about based on these experiments and what did we find. So that's why we never use the word truth in science, because we don't know. We may not even reach the real truth, but we know there is a knowledge based on a systematic research. For example, in GE, let us say there is a GE Beet eggplant, what is called Bacillus thuringiensis. It is a cry protein which is toxic to some of the pests, Lepidopteran pests. Let us say you have a BT plant which produces toxin to the certain insects. Okay, now the question is, what is the risk? Okay, what if the beneficial insects in agriculture, like honeybees, they go and eat BT plants pollen? Now it may be toxic to honeybees. So then you have to really see, is it good? Does government should approve this one? Okay, now what you say, is it honeybees exposed to this toxic substance? That means then honeybee has to go and get the pollen. Now, what if you do not express this toxic substance in pollen? It produces in leaves, it produces in roots, it produces in stems, but not in pollen. Then there is no exposure, right? So this is the way you do the risk assessment based on science, based on the toxicity and based on how much pollen is fed by the honeybees. Is that pollen contains any toxicity? All these things are evidence-based. So you say use science risk assessment. But there is a problem. There is a distinction between a pure science and a risk assessment. Let us say you are looking at ragi plant, whether it produces drought tolerance, and you want to know what chemical is involved. You identify that chemical, and you have a control plant without those chemical products, and you have a ragi plant which produces a particular chemical. Then you look at, is it really gives a drought tolerance? You don't put water, and you see at what water level still it produces a reasonable yield. And then you look at heterogeneity, which environment. Then you publish peer review. This is what we do in hardcore science, right? But risk assessment is not. People always misunderstand. Risk assessment is a process where you use scientific data, but risk assessment is not the science by itself. See, let us say somebody comes and asks you, is it ragi a good variety? You say probably yes. Does it really grow well in a particular environment? But I don't know, but you can try, because it's impossible to know everything in a risk assessment. Whatever data is there, you use it. But you cannot do a peer review on a risk assessment you do. For example, a pest, whether it's really pest, what is the extent of its damages, all those things you can do science, you can do risk assessment. But in the risk assessment, if you are asked to give a decision, you have to give a decision with what you know, because you're not doing a hardcore science there. You use science to make that decision. I hope I made this clear. So let us say, in the United States, we started the risk assessment in 1987. What we looked at genetic material. For example, people put material from the bacteria, material from different organism into the plant. We looked at its genetic material at a molecular level, and we looked at whether this genetic insertion makes plant a disease. It's susceptible to disease. For example, we have an apple variety. What they have done is they reduced a chemical called PPO. If you reduce this chemical, when you cut apple, it doesn't become brown. And there is a quality. But the question is, that chemical is also important for disease resistance. You looked at by removing this chemical, are you making plant more susceptible? Okay. And potential impact and non-target, like I gave in honeybee example. Would my GE plant harm beneficial insects in agriculture? Then we say no, you cannot. You cannot grow if there is harm to beneficial organism. Then we look at whether the GE plant increases weediness. What if somebody wants to use a sorghum, and sorghum is really drought tolerant. But if you put so much that now sorghum becomes weedy, and you will have a hard time getting rid of sorghum volunteers. So that means that you have a weediness problem. Potential gene flow to and weediness to sexually compatible relative. That is the biggest problem whenever you have a weedy relatives of crop plants. For example, sunflower. Sorghum has a terrible weed called Johnson grass. It is one of the worst weeds in the world. What if you make your sorghum drought tolerant and this trait goes to this Johnson grass? Then how do you get rid of the Johnson grass? So this is a problem for agriculture. We look at that one. Potential change to cultivation practices. Like, most of the herbicides are applied before a crop really becomes reproductively mature. But today, most of the companies are putting genes so that you can spray. Even plant is producing grains. Now the question is, now there's a change in cultivation practices. Does it going to affect plant production? And last one is potential for horizontal gene flow. The people were worried when GE, genetic engineering, okay, you are putting antibiotic resistant gene in rice or in corn. What happens? Is it going to transfer to other bacteria and causes resistance in bacteria, making a big human and environmental concern? Well, these are the things we are looking for every plant, irrespective how they were made. Okay, and we approved so far 139 varieties of plants. For example, alfalfa, it's a forage crop. It has a very good forage quality. We reduce the lignin, which is hard to digest for animals. Canola, for example, rapeseed, mustard, which has product quality. Canola with very high, omega, which is human health. Most of the people have to eat fish or fish oil to get omega. Okay, then corn, herbicide tolerant, insect resistant variety, and it's cotton. Recently, one of the universities, Texas in A&M, came out with low gossypal cotton. So now people can eat cotton seed, which is rich in protein. But gossypal it produces in all other parts. Still, it protects plants. Papaya ring spot virus, sugar beet. In United States, 50% of sugar comes from sugar beet, not from sugar cane. It is herbicide tolerant. And there are color change in rose. There is no black rose earlier. Now a company came out with a black rose. And there is a petunia, pink petunia. And squash, virus resistant, tobacco, product quality. So yeah, plum, you name it. People are trying so many different, varieties, plants. Before coming to, during this presentation, I checked how many plants really tested in the United States. It is more than 100 different species they have tested already. That means this has become a very powerful tool. Okay, some of the notable examples. In 80s, like Hawaii, most of the 90% of the papaya in the United States produced in one state, Hawaii. There is a papaya ring spot virus and completely eliminated papaya. And there is no source of resistance. And they came to United States Department of Agriculture asking, they really want to use this technology, it's RNAi technology, so that they can produce a resistant papaya. And we approved within no time, probably 1 year. And today, all the papaya, there is a huge papaya industry in Hawaii. What it says is you have a solution, agriculture solution, when you do not have anything else available in conventional breeding. Similarly, we have this quality improvement in soybean, making an unsaturated fatty acid. You know that saturated fatty acid causes heart disease and diabetes. Now, we have 3 or 4 big companies produced unsaturated fatty acid in soybean and canola. In canola, which is rich in this omega, which has a tremendous health benefit, and also especially for vegetarians who don't want to eat, who do not eat fish or do not drink fish oil. And last week, I approved 2 varieties. One is USDA approved commercial cultivation of a corn variety that produces phytase. Like phytase is the biggest problem in an animal manure. So a lot of phosphorus is left in the soil, which is not good for the environment. A company came out. It's a very small company. What they have done is put a phytase enzyme in corn, which converts phosphorus. So that way manure does not contain a lot of phosphorus. And also the forage quality is really good. Similarly, there is another Canadian company asked one of the varieties of their apple, non-browning. What they have done is using a technology, RNAi, that RNA interference technology. They prevented the production of one of the enzymes. It's called PPO, polyphenol oxidase. Because of the removal of enzyme, now if you cut apple, you keep it, it doesn't become brown. So there is a product quality. Now kids really love this apple because it doesn't become brown when moms cut the apple and sent it to the school in their lunch boxes. And also, there is a use of plants to produce pharmaceuticals. There is a company, more companies, but this one example. They produce human lactoferrin, isozyme, and serum albumin rice. Remember, rice is not the natural, a cultivated plants in the United States. But this company produces rice in the middle of nowhere, in the very cold places, in summer, thousands of acres, and produces human, insulin, lactoferrin, lysozyme. And I checked, its street value. 100 acre of rice, they can produce a $1 billion worth, of these pharmaceuticals. It's so easy for them. Then recently I issued a permit, like for example, this is a very interesting example. Army, US Army came to us asking for, we have a plant, what we have this range, it's called military range. They practice all this war exercise, and that soil is contaminated with all these chemicals used in bomb, like RDX and TNT. They are very toxic. They are in the soil, they leak to the water and contaminates, drinking water. But cleaning up this is $1 billion question, so expensive. Then they, scientists came out with a neat idea. They found an enzyme, in bacteria. If you put it in the plant, if you grow that plant on this contaminated soil, it sucks up all this RDX and TNT and makes and converts into a non-toxic substance. Such a great idea. Very limited. It is one of the grass species, native grass species called switchgrass in the United States. So look at the environmental benefit of modifying plants. So, and also producing pharmaceuticals. which is so easy in plants compared to in animals, because you do not have a cross-contamination of proteins. And I think biggest example, which I'm really proud of is this citrus. In last 15 years, in Florida, they produce billions of dollars worth of citrus in orange juice and orange fruits. And in 15 years, the yield decreased by 75%, simply because of one disease like citrus greening. It is there in India also, citrus greening. So almost, they have to remove all the orchards because there is no natural defense in citrus for this bacterial disease. But a company came out with a neat idea. What they have done is they took a defensive gene from spinach, another plant, put it in citrus tristeza virus, which causes disease. They removed all the virulent genes, put this gene from spinach and inoculated, bud inoculation to all the citrus plant. Now it provides resistance to this disease. So such a neat idea. It took 3 years for me to approve this one, but this is how you solve an agriculture problem when there is no solution exists. That is the beauty of this genetic engineering, which is just another plant breeding technique. There is nothing new about it. So what did we learn? We said science, right? You acquire knowledge in science, but how do you make use of it? You don't, use the same thing. You always want to improve, right? Regulation is always, it should change, and it should based on how much you know. You don't want to over-regulate because it costs business, That's why in 30 years, we have experience, working on so many plants, and we have approved 100,000 field trials, and we 139 plant varieties are commercialized. And the question is, what did we learn? Okay, this is what we learned. Okay, because of our regulation, look at how much it costs to produce a variety of plant in the United States. It takes 10 years. It almost costs $100 million to produce one variety, which is astronomical. Okay, earlier, if you look at the Green Revolution in India, all the universities, all the research stations came out with so many varieties, high-yielding varieties for the different agroecological condition. They did not spend millions of dollars. And there is a food security in India. But remember, because of this regulation, we made it so expensive for farmers and also the researchers and the universities that only, nobody can really produce except big companies like Monsanto, Bayer, now Syngenta, BASF, and Pioneer. No other people can really come out with these kind of varieties. That means it is because of over-regulation we did. What did we do? Another lesson is only big biotechs could afford GE crop improvement. Small developers and universities completely left the field because they cannot deal with it. Who is going to give them so much money? Then we realized, okay, we are over-regulating. We are really killing innovation in agriculture. So that's what we learned. Okay, just this cartoon says, what is happening is we are regulating the technology because something is GE, it has to be regulated. If it is recombinant DNA, you have to regulate. If you produce the same thing using conventional breeding, you don't need to regulate. So then we thought it's some kind of hypocrisy what we are doing here by regulating the same thing if it is GE, but not it's conventional. So what we look at is ultimately it is not the molecular biology we are worried about. The safety issue is always the phenotype. If there is a risk comes from the phenotype, not just from the technique we use to create that phenotype. Okay, lessons learned. Environmental risk. Risk to plant or the environment comes from the plant traits or the phenotype, not because you put a gene there. GE techniques are not inherently risky. See, we regulate because somebody used a genetically engineered technique. Technique doesn't have any risk. It is how you used it. It's like using knife. You can cut vegetables, or you can stab somebody with that. Just like, it's just another technique. Regulation should be then product-based, not which process you used. That is, if somebody produces a plant with more toxicity, so you have to regulate that plant phenotype, not because somebody used the technique. You can say produce a plant with toxicity, including using conventional breeding. So the process should not be you should not regulate process. Then your regulation should fit for the purpose, strike a right balance between risk and oversight. See, when I have power, I really want to enjoy that power. I want to regulate more and more. That really doesn't help agriculture, doesn't help developers. Our regulation should really meet what is the need. Is it really a risk? 90% of the time, there may not be any risk. Then why are you regulating? Okay, now we revised in 2020. We revised regulation after 3 decades of experience. Advances in science and technology have changed so much in agriculture. Now we want to establish a clear, consistent, science-based and risk-based regulatory framework for biotechnology and provide regulatory relief and better focus on regulatory resources on areas of risk. That means